Okay, so we're going to go into cellular respiration. We're going to start with a process that occurs outside the mitochondria. It is called glycolysis. During glycolysis, small amounts of ATP are generated using what's called substrate level phosphorylation. Substrate level phosphorylation is where we are going to use an enzyme to put a phosphate onto a molecule to make ATP. So when we talk about ATP versus ADP, remember ATP has 3 phosphate groups versus ADP has 2. And so what we're going to end up doing is pulling the phosphates off of our substrate and phosphorylating, sticking them onto ADP to make ATP. So this process is called glycolysis. It's a lot more complex than what we're showing here, and each step has a specific enzyme and step that goes through it. This is a progression of steps that occur. And so we know this is like found in pretty much all living organisms do glycolysis, which is why we believe that this is one of the first processes of metabolism to obtain energy. In this particular pathway, we do not use oxygen. So, and again, it doesn't take place in the mitochondria, which we know all prokaryotic cells do not have membrane-bound organelles. And so I'm just going to kind of break this down into some simple steps. And hopefully I have space here. So we're going to take glucose. Glucose is 6 carbons. 1, 2, 3, 4, 5, 6. Now there's more than just carbons on this, right? It's C6H12O6, right? But we're going to follow the carbons. And the first thing that we're going to do is we're going to have what's called our investment phase. And we're going to actually, and I'm going to shorten this a little bit, some of the steps because I'm kind of running out of space here. We're actually going to invest 2 ATPs, and then we'll get 2 ADPs out. And then from that, we end up getting glucose 1,6-biphosphate. So we stick those phosphate groups on that molecule. All right, so like I said, this step here is referred to as the investment phase, because we have to put a little bit of energy in there. What happens when we put those phosphates in there, it puts a lot of stress on the middle of that molecule. And so that molecule ends up splitting into 2, 3 carbon molecules. Let's stick our phosphates back on there. In there. All right, so once we do that, we are actually going to do another step to this. And in this step, we're actually going to do 2 things. And I'm going to have to kind of go over to the other side. First of all, we're going to put some independent phosphate in there. We're going to put that in. And we're also going to oxidize the sugar. So we're going to go from NAD+ to NADH. So we're going to pick up some electrons. That is really important. What we're doing is we're pulling off the electrons in the form of hydrogen. Ooh, let me do this. Make my H a little better. So that we can do work with it later. And that work is going to generate a lot of ATP. And so then I'm just going to kind of carry on over here. And then we're going to have our, I always call them, I refer to them. I refer to this one as my big poop. And now I got 2 little poops. So I got my big poop, I break it down to a poo and a oop, and then I have 2 poops. And then the next step, we're simply going to be pulling those off. Oh, and let me stick a number in here because we have 2, right? We're going to have 2 of these and 2 of these and 2 of these because we have, right? We have 1, 2 molecules. And so when we finish this process, we're going to pull that off and we're going to end up with 2 molecules of pyruvate. And we're going to pull those phosphates off, and we're going to actually take 4-ADPs and get 4-ATPs out. So as we get those ATPs out, we made 4, but remember we invested 2. So our profit margin is only 2 ATPs in this process. So it doesn't make a whole lot of energy, but it sets us up for the next part of metabolism. All right, so here, this is kind of showing you our input. We put in glucose, we get out our pyruvate, we put in we invested our 2 ATPs, and then we also put in our NAD plus. Remember, I did that in this color. So we got out these, we're going to use these later. And then we made 4 ATPs. But really our profit margin, because we use 2, is only, so our profit is 2 ATPs in glycolysis. And then there's a little video for you to watch on that process. After we get through glycolysis, we are then going to enter what's called the citric acid cycle. So we're going to actually go into the mitochondria. The first step we have is oxidation of pyruvate. It has many words. This one's also called the intermediate step or the preparatory reaction. So this has 3 different names. So in this progression of things, what we're doing is we're taking pyruvate, we're going to pull off that CO2. We're going to stick it on an enzyme here, and we end up with acetyl-CoA. We're also going to pull off, notice I'm also reducing my NAD plus to pick up more electrons. So we are, again, stripping our 3-carbon sugar, pyruvate, of its electrons, and we're getting rid of the CO2. The CO2 is what you are exhaling. So that's our first carbon that we've gotten rid of. Now, remember, we have 2 of those. So we have 2 pyruvates going through. And here's another picture of this. So we have our pyruvate, coenzyme A. We pull off our CO2. We are then going to take our acetyl-CoA, and acetyl-CoA is then going to go into the citric acid cycle. All right, so I'm not going to draw the intermediate step, because I mean this is pretty clear what we're doing. The citric acid cycle, though, is kind of confusing, so I will be going through that in a little bit more detail. So the citric acid cycle is called that because, we can see citrate, which we first said our product in this series of chemical reactions was citric acid, so we called it the citric acid cycle. Krebs is the person who came up and discovered all these steps, so it's often referred to as the Krebs cycle. So the first thing we're going to do is we're going to have our acetyl-CoA come in and drop off our 2-carbon molecule here. And so we're going to have a 4-carbon, I hate it when it does that. We're going to have a 4-carbon molecule hook up to a 2-carbon molecule. This is 4 carbons. Let's see if I can do this without going to 6-carbon molecule. And then we're going to continue through our progression of steps, which I'd rather draw out than to use this picture. So I'm going to get to that here. Okay. And I left my short notes at school, so I have it pretty well memorized, to be honest. So we're going to have our circle. We have our, we are going to have, I'm like totally getting Calvin cycle and Krebs cycle mixed up at this point in time in my head. We have ribulose biphosphate, RUBP, which is a 1, 2, 3, 4, 5 carbon molecule. See, I told you I had those mixed up. Let's try this again. We'll be doing this next chapter. You'll see why it's so confusing. We have oxaloacetic acid. Let's try that one. Oxaloacetic acid, which is a 4 carbon molecule, and it's going to hook up to our 2 carbon molecule here, our 2 carbon, which is our acetyl-CoA. So we have 2 carbons coming in here, coenzyme A, and they're going to hook up and they're going to form a 6 carbon molecule called citrate. All right, so 4 plus 2 gives me 6. And then what we're going to do is we're going to oxidize our sugar. So what we're going to do in our first step is we're going to get rid of 1 CO2, minus a carbon. We are also going to and that technically should be NADH plus H plus, but I'm going to run out of space, and so I'm not going to put the H plus in there, but we do. So again, we're exhaling the CO2. We went from 4 to 2, that gives us 6. We're now down to 5, a 5 carbon molecule, and we're going to repeat this step to get us a 4 carbon. So we're going to get rid of a CO2. We're going to reduce our NAD plus, get NADH plus H plus. We are also in this step, we have enough energy to make a molecule of ATP. And then we're going to continue around. And then we're going to pull some more electrons off. We're going to have a different type of electron carrier called FAD. And so we're going to then go from FAD to FADH2. You can see we're picking up those hydrogens again. And so as we pick up those hydrogens, we're going to reduce that electron taxi cab. And then we're going to pull off some additional, we're just going to keep stripping all the electrons off this sugar that we can possibly, and I have room, so I'm going to put it here. All right, so again, the main point is we are oxidizing the sugar. Another way of saying this is that we are harvesting electrons. And you might think, well, why are we doing that? And we're going to get there. So we are harvesting electrons. So right now we have gotten rid of, we brought in our 2 remaining carbons. We got rid of them. We've exhaled them. So we've completely burned pyruvate, which was, again, this is going to go around twice, once for each pyruvate. And so the carbon, the sugar that you ate, we have stripped off the hydrogens. We've gotten rid of the carbon and the oxygen as waste. And so here is our combined stages. So we had 4 ATPs from glycolysis, but remember, we have a total of 4. Remember, only 2 came from glycolysis, and we had 2 from citric acid cycle. We had our 6 CO2s. Remember, we got rid of 2 from the oxidation of pyruvate and 4 from the citric acid cycle. We're up to 10 NADHs, 2 from glycolysis, 2 from oxidation of pyruvate, and 6 from the citric acid cycle, and then our 2 FADs. So this is kind of a summary of all the products that we have made at this point. The important ones we want to focus on would be the ATP, which again, when we get done with this, 1 glucose molecule is going to give us around, right? That was the old school around signal. around 36 to 38 ATPs. We've only got 4 here, but this is important and this is important. We're going to cash them out. We're going to cash them out in oxidative phosphorylation. So remember, we haven't used oxygen yet, right? You understand that you've exhaled your CO2, but why do we inhale oxygen? And this is where the oxygen comes in. So oxidative phosphorylation has 2 parts, the electron transport chain and chemiosmosis. Both parts occur in the inner membrane. So we're going to be in the inner membrane. Right now, the Krebs cycle was in the matrix in the middle. So we're going to be in the inner membrane for the next few steps. And this is where most of our ATP is going to be made. So we can see here, this is that inner membrane. Oh. Let's see if I can type over here. Inner membrane. And so what's going to happen is with our electron carriers, once they're full, we have to unload them or we can't keep going on in our steps. So we need to have a place that we're going to drop them off. And so we're going to drop them off. This part here is referred to as the electron transport chain. And so as we drop it off, that electron is going to get passed. Now, every time it's passed, it's doing work. And the work that we're doing active transport. We are transporting protons, H pluses, into the intermembrane space. So as we are pumping this up, we are making a concentration gradient. We have a high concentration of protons up here, and we have a low concentration of protons in the matrix. And so what's going to happen is they're going to want to diffuse, right? That's passive. They're going to want to diffuse through this protein that is called ATP synthase. And as it goes through ATP synthase, it will do work. That kinetic energy will do work. It'll convert that kinetic energy to chemical energy, and it's going to phosphorylate ATP. The problem we have is if we are stopped here with our electrons, we cannot continue to pass. We get a traffic jam. And so we can't continue to pump protons to make ATP. So we need some molecule to pick up our electrons. And as we have studied, oxygen is highly electronegative. It wants electrons. And so we're going to end up using oxygen right here to pick up our electrons. So this part here is called chemiosmosis. This part here is ETC. All right, so I'm going to draw this here. Let's do this again. So this is my inner membrane. All right. You could say the outer membrane is up here. This is all in the, I'm just going to put IM for inner membrane and I'll run out of space. So this is in the mitochondria. Down here would be our matrix. We are going to have 3 proton pumps. I'm squishing them here together here. And as our NADH, remember it's plus H plus, comes through, it's going to drop those electrons off. And those electrons are going to get passed I went too low with that one. And I kind of was trying to go downhill at a steady slope because every time it does work, it's losing some energy. And the work it's doing is it's pumping those protons up into this intermembrane space. At the end here, you're going to have oxygen plus our H pluses combine with our electron to make water. metabolic water, which you exhale as well, right? And so we're going to do this. This here is ETC, electron transport chain. Now, remember, we also have another electron carrier. We're going to drop those electrons off to our FAD. So we're going to drop those off here. So those electrons are going to join in the party in the second proton pump. And then we have our ATP synthase. And that's going to allow those protons, since we have that proton concentration gradient high up here, low down here. It's going to diffuse, it's going to be passive. And then we're going to go ahead and make our ATP. Now we get approximately 1 ATP per pump. So 1, 2, 3 pumps. So for every NADH, it equals about 3 ATPs, about, as it goes through 1, 2, 3 pumps. Now FAD only goes through 1, 2 pumps. So it's not worth as much. We only get 2 ATPs from that. Okay, so this is where we're cashing out our electron carriers to make ATP. So again, and like I said, the whole thing here, this whole thing is called oxidative phosphorylation. This here is chemiosmosis. This here is ETC. All right, so the electron transport chain, like I said, it's a series of electron carriers that's embedded. And basically what we're doing is we're using those highly energized electrons that we got from sugar to slowly release their energy to do work. So we can see here, this is where NADH and our FADH come in. This is that concentration, highly concentrated intermembrane space. And then we have our water here. So that potential energy is used to pump the hydrogen ions, like I said, in the intermembrane space. We have a hydrogen ion concentration gradient now, and we can use that for chemiosmosis. And then at the end of the line, we have oxygen, which is going to pick up those used or discarded electrons that are now at a ground state and have no more potential energy. And then we have chemiosmosis here. Again, that's with that ATP synthase. So we're going to generate, again, that kinetic energy is going to be used to, we're going to convert it into chemical energy, into that chemical bond between the phosphates to put that third phosphate together on ADP. And so here is a video that you can watch on this process. You can see here with our Krebs cycle in the matrix, and then our progression of our ETC, our chemiosmosis, which are all parts of oxidative phosphorylation. Well, other than the Krebs cycle, but you can see where those products that we made are being fed into the electron transport chain. All right, so I hope that presentation was helpful for you. And if I have also some other videos in Canvas to help you with understanding this process. I know it's a difficult one, but it will get a little bit easier, especially when we get to photosynthesis. It's basically this in reverse. So once you understand these concepts a little bit easier. I know these 2 chapters are going to be difficult. All right, so if you have any questions, please feel free to ask me, and I will see you in lab. All right, bye.